Measuring genetic diversity. Why is genetic diversity important? Well, this is a review from the last lecture, and that is because A, genetic diversity is required for populations to adapt to environmental change, and B, the loss of genetic diversity is usually associated with inbreeding and an overall reduction in reproduction and survival. Well, there are a couple ways that we talk about and measure genetic diversity. We kind of think of this in generally, we're looking for polymorphism. And polymorphism is the presence of more than one allele at a locus. So if something has many alleles, we call it polymorphic. And if it has few or one allele, we call it monomorphic. So let's take a look at a gel to kind of go over this. So here we have these gels that we've been looking at, and I've colored the different alleles, different colors. So you can see there's 4 alleles, blue, purple, red, and green. And this indicates that this locus is polymorphic because there's more than one allele. Now, in contrast, here is a gel that shows us a monomorphic locus where we have a single allele for every sample. So this is monomorphic. So we actually use different metrics for measuring genetic diversity depending on if we're looking at DNA sequences or if we're looking at genes within a population. We're going to start talking about what we do when we are looking at sequences. So here we have 3 different sequences. They are from different individuals and they are aligned together. So all of the common nucleotides match up. And then you can see the blue and the black ones represent areas where they're different between the sequences. So to measure genetic diversity in these types of sequences, we look at nucleotide diversity. And nucleotide diversity is the average pairwise sequence difference per nucleotide. There is a equation that, so we actually use different metrics for measuring genetic diversity depending on if we're looking at DNA sequences or if we're looking at genes within a population. We're going to start talking about what we do when we are looking at sequences. So here we have 3 different sequences. They are from different individuals and they are aligned together. So all of the common nucleotides match up. And then you can see the blue and the black ones represent areas where they're different between the sequences. So to measure genetic diversity in these types of sequences, we look at nucleotide diversity. And nucleotide diversity is the average pairwise sequence difference per nucleotide. So this is the equation that you can use to figure this out. Let's kind of talk through this. So nucleotide diversity equals pi, which is the sum of these 3 factors. And this one right here is the differences between each pair of sequences. And then this is multiplied by the frequency of each of these 2 sequences. So it corrects for the amount of times that you see that allele in the population. So the first thing, let's do this. The first thing that you need to do is figure out all your pairwise comparisons. So here we go. You can compare sequence 1 and 3, 3 and 2, and 1 and 2. And if we start with looking at this factor for the first pair, we can see that when we look at the 2 sequences 1 and 3, they have only one place right here where they're different. So that means that 1 out of 10 nucleotides are different, which gives us a frequency of 0.1. So we'll put down 0.1 there, and then we'll take a look at this other factor. So this xi is the frequency of this allele. So the way you do this is probably given to you, or essentially what you do is when you're sampling a population, you sample it over and over, and then every time you get that same allele, you mark it in, okay, that's this allele, that's the number of times I got that allele, this is the number of times I got that allele, and then you estimate the allele frequency. So here, this is given to us. So 78% of the time, we pull out a number 1 allele, 19% of the time we pull out a number 3 allele, and 3% of the time we pull out a number 2 allele. Okay, so then in this comparison here, we're looking at 1 and 3. So we take the frequency of 1, this 0.78, and we multiply it by that. And then we also multiply it by this 0.19, which is the frequency of 3. So that's the first set, and then we have to do this for each pairwise comparison. Here we move that over, then we compare 3 and 2. So when we look at 3 and 2, There's 1, 2, 3 differences, which is 3 out of 10 for 0.3 frequency. We have that here. And then we take the frequency of the 3 allele, which is 0.19, we put it here. We take the frequency of the 2 allele, which is 0.03, we put that here, and multiply it together. And then we're summing these two, and we do this again for the third one. Here again, you can see that between 2 and 1, there are 3 differences. Frequency of 0.3, so we put that here. We take the frequency of the first allele, which is 0.78, put it here. Frequency of the second allele, which is 0.03, put that here. So then we multiply these first, and then sum all of these, and we get the nucleotide diversity, which is 0.02, or 2%. So what does this actually mean? This result means that for any 2 sequences chosen randomly in a population, 2% of the nucleotide sites are expected to be different. You have to think about when we are going to use this, when we're using sequence data. Well, if we think back to what we know about different types of markers, we use sequence data when we're looking at mitochondrial DNA and comparing different sequences. We also can use sequence data when we are looking at AFLPs. So if you remember, AFLPs are where you have that restriction enzyme that cuts a very specific code. So you can think of it as if it cuts that specific code, then it has those nucleotides, and if it doesn't cut it, then it doesn't have those nucleotides. So when you compare, here we're looking at these places where there are these arrows, these are where there are differences between populations. That means that these samples over here don't have that nucleotide and these do. So that means that there's a difference in nucleotides between this group and that group, and this is true for these individuals too. So you would then use those numbers in your calculations of nucleotide diversity. Okay, we just talked about measuring differences between sequences, but you use different genetic diversity metrics when you're measuring genetic diversity for genes in a population. So here they are. When we're looking at genes, we look at several metrics. The first one is the proportion of polymorphic loci. This is the number of polymorphic loci divided by the total loci. And another one is allelic diversity, which is the average number of alleles per locus. Okay, so what does this mean? Let's look at a gel here. So here we have 2 different loci. They are blue, one is blue and one is purple. So when we're calculating the proportion of polymorphic loci, or P, we take first the number of polymorphic loci. So we can see that the purple locus is not polymorphic and the blue one is. So we have 1 out of our 2 loci that is polymorphic, and you divide that by the total. So your P here is going to then be 0.5. So for allelic diversity, you look at the average number of alleles per locus. So if we look at the blue locus, we have 3 alleles, 1, 2, 3. And then for the purple locus, we have 1 allele. So that would be 3 plus 1 divided by 2, because that's the number of total loci we have. So 3 plus 1 divided by 2, and that's 4 divided by 2, so our allelic diversity ends up being 2. So heterozygosity is also a way to measure genetic diversity, because if you think about it, when you have a heterozygote, here we go, we have heterozygote blue and green as opposed to green and green. If you have a lot of heterozygotes in a population, you have a lot of different chances to have different alleles. You can see here in this group we have a lot of more different colors than we have in this group. And so therefore when you measure heterozygosity, it can tell you about the genetic diversity of a population. And the metric we specifically use is something called average heterozygosity, and this is the proportion of heterozygotes averaged across loci. So let's measure this in a gel. So here you have the same gel, you have 2 loci, the blue and the purple. If we measure the number of heterozygotes in the blue, you can see that we have 1 heterozygote, 2 heterozygote out of a total of 4. So that would be a heterozygosity for the blue locus of 0.5. And then for the purple locus, it would be 0 out of 4. So that would be 0. And then you take these two and you average them. So that's 0.5 plus 0 divided by 2. You get 0.25. So the average heterozygosity for this population is 0.25. we can compare the allelic diversity and the average heterozygosity in different populations and tell how genetically diverse a population is. So let's take a look at this table. This is pretty interesting. Here we're looking at a group of threatened species here on the left compared to a group of non-threatened similar species on the right. And if we look at the allelic diversity here for both of them, you can see that in the threatened species, the allelic diversity is much lower than in the non-threatened species. Similarly, if you look at heterozygosity, you can see that the heterozygosity is lower in the threatened species than the non-threatened species. And so this indicates that there is more genetic diversity in these non-threatened species. So you would calculate these genetic diversity metrics related to genes when you're using markers such as microsatellite or SNPs, because here you're going to have multiple loci, you're also going to have different alleles, and you're going to be able to figure out whether or not they're heterozygote, because in both of these they're codominant, so you can see the alleles from both your mother and father. So, enhancing genetic diversity. How would you then enhance genetic diversity if you were charged with managing a population? There's 3 general ways you would do this. First, bring in new individuals. Second, would be maintaining habitat connections between populations. That way individuals can move themselves, freely move between populations, and they can bring in new individuals and change the genetic makeup of populations. And the third one is if you had maybe a captive colony, or even if you had a small population in the wild, you would then try to actively manage matings. And this way you would try to bring in new diverse alleles and enhance the genetic diversity.